Good evening, everyone, and welcome to myDNAhealth's webinar on the genetics of celiac disease. Thank you for joining us this evening. So, just a little introduction to our presenters. We have myDNAhealth Scientific Officer, Dr. Eve Pierce, and from Cytoplan, we're delighted to have Clare Daley. And let me tell you a little bit about them before we start. Eve is experienced in biochemistry and genetics, and is a practicing clinician. She has over decades of genetics and medical research experience. She gained her PhD in medicine from the University of Wales College of Medicine, later working in a genetics laboratory as a research fellow at the University of Southampton. So loads and loads of genetic experience. During this time, Eve also published a number of research papers in medical journals and has also trained medical students. Eve's passion for wellness and nutrition led her to complete her ION diploma in nutritional therapy. So you'll be in good company this evening with all practitioners who are listening in. Her clinical interests include genetics and epigenetics, nutrition and dietetics, nutritional biochemistry, and cancer research. Now, a little about Clare. Clare worked in private practice for a number of years and for Nuffield Health. She joined Cytoplan in September 2015 as an in-house nutritional therapist, where she produces and delivers nutritional education, writes and edits blogs, and supports practitioners and their clients. Clare has a particular interest in brain health and runs Cytoplan's Action Against Alzheimer's program, which is an amazing program, and myDNAhealth certainly supports this program. Okay, now let's just go through the agenda. Okay, so it's in two parts this evening. Eve will be covering the first part. She'll be going through celiac disease definition, diagnosis, and the mechanism. She'll introduce the genetics and testing the HLA locus. She'll cover the approach that myDNAhealth takes, and there'll be a few scenarios and case studies to share with you. We'll then take questions from Eve, and then we'll hand over to Clare. And Clare will discuss factors affecting brain-gut axis, including gluten sensitivity. And we will then take questions, pose questions to Clare. So please, again, post those questions up in the chat box. Okay, so before we get started, we have a quick poll. So Sylvia, can you load the poll, please? Okay, if you can answer and make your choice. So the question is, do you generally avoid gluten, or are you on a gluten-free diet? And we'll just leave it live up there for a few minutes, and I'll be muting myself and handing over to Eve. Thank you.
Hi, thanks for that. That was really interesting. Thanks, Bernie. Good intro. The poll was really interesting. It looks like a lot of you are following a gluten-free approach, but then on the flip side, there was some of you that feel that you're totally tolerant with gluten. So we'll see how that fits in with the genetics. I know that I'm quite tolerant of gluten, and I also know that my genetics are quite far away from celiac. So we'll see if that has some sort of impact. So I'm going to start off by defining celiac disease. I'm going to look and just run through what this is. So the definitions tend to define it as an immune-mediated disorder, and this is through a response of T cells in response to a gluten ingestion, and that's in genetically predisposed individuals. So it's quite a heavy definition there, but it's characterized by a variable clinical presentation, often with damage to the intestinal wall, which is seen as the breakdown of the usual troughs and valleys known as the crypts and villi, which leads to that significant malabsorption and inflammation issues. And as with other autoimmune conditions, celiac disease can present at any point in life. So what causes autoimmune diseases? Is sometimes referred to as a perfect storm of events, which leads to its development. And modern models have identified three main aspects to this, which I'll run through. So there's our genetic component, our environmental component, and our immune function or dysfunction, depending on which way you want to look at it. And if you'd like to learn more about the genetics and epigenetics of autoimmunity, there is a webinar on the myDNAhealth practitioner portal that looks at this in some depth, because it's quite a lot to cover today. So if you want to go back and have a refresher of that, there's a link there to our YouTube channel, which you can go and see that. and get yourself updated on the genetics of autoimmunity. However, we know that celiac is unique as an autoimmune disease, and that we understand both the environmental trigger and how this is regulated by the immune system through the autoantigen known as transglutaminase. So I'm going to go through that carefully today, but the elimination of gluten leads to a complete resolution of the disease. And at present, this is the only accepted treatment of celiac disease and makes it quite unique in the way that we think and work around it. Traditionally, there's 3 points which we must combine to form a formal diagnosis, which medical doctors must follow. And this includes positive serology for IgA transglutaminase, plus a positive identification from a small intestinal biopsy, which isn't terribly pleasant. And the markers for this include a villous atrophy, so that's the degradation of the villi. You can see them completely sloughed off, and enlarging of those crypt spaces, so this is known as crypt hyperplasia. And basically from there we get high amounts of invasion of lymphocytes into the epithelial layer, which is the start of that inflammatory process, which we're all quite well aware of. And these 2 tests then must combine with a clinical improvement following a strict adherence to a gluten-free diet. And only then can we go on to have a diagnosis of frank celiac disease. So I'm just going to talk to you a bit about gluten and transglutaminase. I'm going to explain this diagram to you. So gluten is a mixture of a protein polypeptide strands found in wheat, barley and rye. And one of these, gelandin, has been singled out as being known to cause gluten's antigenic properties in celiac disease. So this diagram is from a paper by Schumann et al., and the reference is there below for you from this year. And it details how the high proportion of glutamine, so one of the amino acids, 30% amino acids within gelandin, leads to the ready modification by tissue glands to glutaminase. So it's a unique property of gluten, really, which allows tissue glutaminase to come up and modify it. So this reaction is where glutamine is transformed to a glutamate, seen here as a change in the polypeptide chain from an L to an E. And this adds an additional negative charge to the polypeptide and basically encourages the binding of certain human leukocyte antigens. These are sometimes called HLAs, the grooves within the major histocompatibility complex, sometimes known as MHC. So there's quite a lot of terminology around this. And those, as we should know, are attached to antigen presenting cells. So those are the ones that really show to the immune system, they present things. And the HLAs are basically the hands that are handing that over. So HLAs are markers located on leukocyte cells, and they help discriminate between host and foreign cells, so ourself and what's not ourself. And in this example, we can see the gelandin fragment alpha-1 binding to the HLA-DQ2, but this could apply to other gelandin sequences and also to HLA-DQ8, but I'll be going into HLA-DQ2 and 8 a bit further in further slides. So if we just think about the mechanism a little bit more and we go back and just sort of get this set in our minds, because it's quite complicated. In this diagram, we can see the proposed mechanism. So when I say proposed, I mean that this is still current research. We're still looking into this quite in depth. There's plenty of research going into celiac disease all the time, but this is quite an up-to-date model. And within the context of the gut wall, and it's been extensively researched for the last 15 years at least. So this was from 2015. And again, the reference is there for you below. But current theory has identified that indigestible gluten fragments cause enterocytes to release zonulin. I'm sure you've all heard of zonulin, but zonulin is a key protein which opens up the tight junctions, which is seen here at 1. And then if we move on to 2, the gluten fragments cross the intestinal lining and accumulate under the enterocytes. So that's the gut wall lining there. And this includes induces the enterocytes to secrete interleukin-15, which is an interesting cytokine known to induce intraepithelial lymphocytes, which are directed against the enterocytes. So already we're seeing some damage within the gut wall there. And at 3, there's this release in response to that of the tissue transglutaminase 2, that's its full name, and then that deamination reaction of the gelatin, which basically makes them more adapted to and ready to engage with the HLA, specifically the DQ28 molecules, which is expressed on the surface of the antigen-presenting cell seen here at 4. So if you have the HLA-DQ28 molecules within your genetics, you're opening up this avenue basically. That's where the antigens are presented to the T helper cells at 5 there, and then activation of the inflammatory cells, which migrate to the intestinal wall, causing that damage. And finally, in addition, we can see that killer T cells can also migrate to other organ systems, such as the bones, brain, and liver, where they can trigger further inflammation and be responsible for a lot of extraintestinal symptoms, as they're known. And I think these are some of the things that we see quite often in clinic with our clients. So given this mechanism, it can be useful just to note the huge range of the extraintestinal manifestations that have been associated with celiac disease. So these are some of the ones that I've picked out of the literature, and the more common ones really seen include anemia, osteoporosis, arthritis, there's peripheral neuropathy, dermatitis, and particularly short stature is seen in children where they basically fail to thrive. However, you can see from this list, the associated conditions can be wide-ranging and very much present as a spectrum. So, epidemiological studies from the 1990s from Europe, North America, and India have estimated that celiac disease affects 1% of the population. However, celiac cases are rising, and this could be down to one of 2 things. So firstly, diagnosis is more reliable now, and more cases are being picked up due to awareness. The celiac iceberg here is quite traditional when we're talking about celiac disease. It's quite well used. Here illustrates a spectrum of presentation we've looked at. However, awareness is increasing, particularly those extraintestinal manifestations we talked about. And even in latent cases with normal mucosa, they have genetic susceptibility and positive serology. Secondly, there's a mounting gastrointestinal research focusing on the leaky gut, including how our microbiota affect our gut integrity. Is an increase in leaky gut leading to more cases of celiac disease? I think within the research, we're far from knowing the answers to these questions at present. So we've looked at the environmental trigger of gluten and possible leaky gut microbiome influences, and then the following immune dysregulation, starting with the tissue transglutaminase 2, leading to that activation of killer T cells and a wide range of health conditions. But so far, we haven't really touched on the genetics. So I'm just going to go into that now. So let's have a look at the overall genomics of the case, so the overall picture. Studies have consistently identified that if you have a first-degree relative with celiac disease, you stand a 7.5 chance of developing this also. If you have 2 first-degree relatives, this jumps to 17%. And as with most genetic overview studies, the gold standard is to study cases in identical twins. So these are monozygotic, and these have a 75% chance of both having celiac disease. So this shows us again that celiac disease does not have full penetrance. From this information, it can be identified that inheritance is in an autosomal dominant manner. And if we move on to look at the data from the genome-wide association studies, where DNA changes are looked at in groups of celiacs and non-celiacs, and then statistically analysed to identified areas of the genome associated. So it's a good way of getting a whole overview of someone's genome within groups. We see that there are 43 common areas which have been identified as contain containing up to hundreds of single nucleotide changes, and that so within celiac disease, and those are all related to celiac disease. So that's a lot of haystack to look through to drill down the definitive genetics cause of celiac disease. And obviously we are far from that at the moment, but interestingly, 90% of these loci, so areas, are identified in the non-coding regions of the gene. So that's areas that are not actual genes, they're in between. And those areas which have not been identified basically by the Human Genome Project as coding for genes in order to express proteins. And this information may seem a little overwhelming, but is very much in line with current understanding of the genetics of all other autoimmune diseases. And it explains why no reliable genetics tests exist for diseases such as type 1 diabetes or psoriasis. So fortunately for us, celiac genetics is a little clearer, and the reason for this lies in its unique immunopathology, which we've discussed, and it is affected by those specific human leukogen and antigens, those HLA variants expressed on antigen-presenting cells, and these are estimated to contribute 25 to 40% of the genetic variation of celiac disease. And within autoimmune genetics, this is a very large amount and very significant. So the fact that 40% of Europeans are positive for HLA-DQ2 or 8, but only 3% then go on to develop celiac disease shows that these HLA variants are necessary, but not totally sufficient for disease progression. However, only 6% of celiacs do not have either of these HLA-DQ2 or 8 markers. So let's just have a look at this HLA locus in some more detail. The HLA genes are known to confer risk to several autoimmune and inflammatory disorders, and they're located in the major histocompatibility complex region on chromosome 6. So this region is 3 million nucleotides long and highly polymorphic, which means it's subject to a lot of genetic variation and is essential in our immune response. So the major histocompatibility complex is part of this mechanism, which allows our immune system to distinguish between self and non-self, as I said before. So going into the details of this mechanism is beyond the scope of this webinar, but if you feel you need a lecture, why not try this AK lecture on the link below, and that will give you a good overview for you to further your knowledge. The MHC complex is made up of 3 different classes, which each have their own subclasses, of which class 2 contains the subclass we're interested in here. So we're focusing on the HLA-DQ section. The HLA-DQ locus, so that's the area of the genome, represents 2 small gene areas which lie next to each other, and these are known as A and B, both of which contain many different genes. The DQ antigen protein is composed of 2 polypeptide subunits, so from these genes we have 2 proteins made, one from the A class and one from the B class. And in this diagram, we can see that an immune cell, and within its nucleus is indicated the HLA-DQ locus. So the green area is A group and the B is in blue. In order to make an MHC protein, which is then expressed on the cell surface, the cell must express proteins from both gene A and B, which then combine to form the MHC molecule. The different HLA-DQ proteins we express is dependent on both the different variants that we have inherited from our mother, see here on the maternal strand, and from our father, the paternal strand there, and on the different combinations which can be produced by these. So this is a diagram that I've just put together to help you try and understand and to represent the combinations of just 2 DQA genes and just 2 DQB genes. And our genetics thereby determine whether we have the capacity to express the known combinations for HLA-DQ2 or 8 found in celiac disease. And this is what we would be looking at when we're looking at your individual genetic variation. So in order to understand your genetic makeup and understand if you carried copies of DQ2 or 8, it was traditionally necessary to sequence large areas of chromosome 6. This is highly laborious and costly, and it's very much not suitable for high throughput screening. The Human Genome Project uncovered our genome and allowed the identification of our common single nucleotide polymorphisms, or SNPs, and these are what we use within the myDNAhealth approach to look at our genetic variants. From this, a powerful method has been identified within the research to use certain SNPs, or genetic variants, which represent regions of our genome. And these are known as tag SNPs. And basically, each SNP is representative of an area of our genetic makeup. And a SNP tagging method has been identified to detect HLA risk alleles for celiac disease. In 2008, it was Monsieur et al. Described 6 tag SNPs to cover HLA risk alleles for celiac disease, which included 2 negative prediction SNPs. These SNPs have been tested in different populations. So far, there's evidence in the UK, the Dutch population, Spanish, Finnish, Hungarian, Italian, and Iranian. And it's been found to be highly specific and sensitive. The 4 different haplotypes are looked at using 3 SNPs to cover DQ 2.2 and 1 for 2.5, 7, and 8. So HLA DQ2 is covered both by 0.2 and 0.5. So we're covering both areas in both the cis and trans orientations for those of you who have a little bit more knowledge about celiac genetics. So I'm just going to talk you through the myDNAhealth approach. Basically, we've taken this method and developed our own algorithm to look at all the 6 of those 6 tagging SNPs to provide an indication of genetic susceptibility to celiac disease. And this can give us up to 95% of the picture based on the research that has been done. And it is based on that research identifying the SNP combinations in celiac populations. So the first clinical scenario I can give you, this is a clinical scenario where we have results for the genetics above, and then below there's an environmental questionnaire that the client can take to determine their epigenetic side. So we have the genetics and epigenetics done for you. And in this case, looking at the genetics, you get the result, and then that is fed into an algorithm within myDNAhealth, which in this case would indicate high risk for celiac disease. And then looking at the environment, you can see also here that the questions will be indicating a higher risk as well. So if this was a scenario, it would be wise maybe to send this client for further investigation for diagnosis of celiac disease, of which you could do through their medical practitioner, but remembering that the diagnosis is only established by that positive transglutaminase IgA serology, the duodenine histology, and showing those pathological changes that we talked about, and then that clinical improvement after removal of gluten from the diet. So that's all very well and good, but what happens if then that patient or client comes back as a negative still? So we're just going to look at a scenario here, which might give us some more help on that. So clinical scenario B was a male, age 53. He had weight gain despite not overeating. So it's both BMI and waist measurements were in the unhealthy range. Persistent and unexplained abdominal and gastrointestinal symptoms, so regular abdominal pain, all things that we see quite often in clinic, bloated stomach, lots of excessive wind, feeling sick, sweaty and low energy as well, and some skin rashes there. So a brief history, the patient has had stomach problems since childhood, at least remember from 12 years old, so that's a long history. Regular visits to the GP, but none were able to provide diagnosis, so plenty of tests done there. Lots of things that we probably would think about in clinic as well, so that's interesting. But currently on a omeprazole medication for acid reflux, we know that that is extremely common and easy to prescribe and give out. So in this case, the test was done. It's coming back as a low risk for celiac disease, but there are HLA 2.2 and 2.5 genetics involved there, and we know that that's significant for celiac disease for the genetics moving forward. But interesting, the patient here has a higher risk given the epigenetics as well, which is indicating that the patient is symptomatic and may be sensitive to gluten, or there can be other gut-related issues to investigate further. So obviously, as a practitioner, you can take all of this into account and look and think, right, well, there's some celiac sensitivity there for the lifestyle questions as well, and your own investigations. And you could consider implementing a gluten-free diet. And the evidence does indicate that gluten-sensitive clients have some of the HLA-DQ28 risk markers, and you can educate your client around this, and then compliance can be quite high if they've got their genetic results as well. Here, so we've got the additional testing. You must do the formal diagnosis, and that indicates that the patient's not gluten intolerant. You can go forward that way, go through the medical professionals, but so really what's help is next is what we can do to help, and we know quite clearly about our gluten-free protocols. We have a lot of knowledge that we can give over about that, which can be quite reassuring, and information to the client they may not have heard before. And obviously if they do start to see improvements, then that's going to help moving forward as well. So this is a case study here of a positive celiac that we've tested. The case history was diagnosed as frank celiac at 14 years old, had short stature and very much failure to thrive. Then once started eating gluten, grew a number of inches in a very short time. So there was positive IgA transglutaminase, obviously, again, a positive intestinal biopsy, clinical improvement following that adherence to a strict gluten-free diet. And he is now being gluten-free for over 30 years. And he said he had no other known issues, and that's what he felt. So if we look at the genetics, he's coming back as a medium risk because he has that HLA-DQ8. So we know that that's important for celiac genetics. That has to be present in 95% of the cases. Now, the gluten sensitivity questionnaire came back as moderate, and that was quite interesting given that he has obviously removed all the gluten from his diet, and therefore his results reflect this. So he noted if he was eating gluten, he would be ticking every box on that, but even having gluten removed from his diet, he still is coming back as quite moderate. So I think I've finished there. I've given you some good clinical scenarios, if there's any questions.
Thanks, Eve. I think that was really very detailed. Lots of scientific evidence there. So we've got a few questions that have come up. So the first one, you mentioned it's 40% of Europeans have HLA-DQ2 or HLA-DQ8, which is associated with celiac disease. Is there a genetic component for gluten sensitivity, including what is known as the non-celiac gluten sensitivity?
So, as I've said, there's links between the HLA-DQ2 and 8 status and gluten sensitivity based on the mechanisms I've described. So those are related to the celiac genes, but there's very little evidence for the genetic origins of the non-celiac gluten sensitivity. And research is really only just starting to understand the mechanism of this, let alone the genetics. But and at myDNAhealth, as you know, we follow a protocol known as the RRA framework, so that ensures that our research is relevant, reliable, and actionable. And at this stage, we wouldn't feel that there's enough evidence to put together a test, and that ensures that we make sure that all our research is robust. But of course, if that changes, we're constantly reviewing the evidence, and we would put in a test around that, but currently no evidence for that.
Thanks, Eve. I've got a second question. I'm going to read it out to you. It's quite long. So here goes. Sales of free from foods is worth over 531 million estimated, with 22% of Brits who are now buying or eating gluten-free products. The growth is also being driven by the health halo of free-from foods and is resulting in a much larger group of users than the limited number of actual or suspected allergy or intolerance sufferers. So the question is, would you recommend cutting out gluten from our diet to minimize our health risks?
Well, this very much depends on the individual case, but the research indicates that those with high, medium risk genetics indicating a higher likelihood of celiac disease, or they have that predisposition there, or of the gluten sensitivity, are more compliant at following a gluten-free protocol, and so that would also be advisable. But those with a symptomatic questionnaire but low risk genetics as well are displaying important epigenetic lifestyle traits. So I would still recommend following a gluten-free protocol in those, but it's really also taking into account all of the information that we do every day in clinic about education around gluten and assessing your patient for leaky gut, all of the symptoms that we know go with that.
Thank you, Eve. And also, I think this just goes back to that scenario that you presented as well. So a last question for you is just going back to some terminology again. Can you just expand on what non-penetrance means?
So penetrance is a term that's used in genetics and medicine, and it's basically the proportion of individuals or people carrying a particular genetic variant, or sometimes it's called an allele, or a gene that also express an associated trait. So in this case, the number of those carrying the celiac disposition, which have celiac disease, is the penetrance. So non-penetrance is the number that carry the celiac disposition, which don't have celiac disease.
Thank you, Eve. Okay, just looking at the time now, what we'll be doing is handing over to Clare, and where Clare will be discussing the factors affecting the brain-gut axis, including the gluten sensitivity. And again, we'll take some questions after Clare's session. And then if there are any other questions, just keep posting them up to the presenters, and we will try and address them as well. So over to you. Thanks, Clare.
Hi, can you hear me all right? I'll assume you can unless I start getting messages up. So hello everyone, I'm Clare, Daley from Cytoplan, and I'm going to talk a bit about, I think I need to change the slide. I'm going to talk a bit about the brain-gut axis and how gluten can be linked to neurological disorders. So the brain-gut axis is a network of autonomous neurons connecting the central nervous system with the gastrointestinal tract, liver and pancreas. And information is transferred in both directions via a number of pathways. So firstly, we've got endocrine pathway, which includes cortisol, which affects the secretion of cytokines, and also cortisol affects the composition and activity of the microbiota. The metabolic route is through short-chain fatty acids from bacterial fermentation in the large intestine, and some short-chain fatty acids have been shown to have beneficial effects on the brain. So, for example, butyric acid. And then others have been linked with behavior changes. For example, propionic acid has been linked with autistic behavior and aggression. Then we've got the neuronal pathway. So the digestive system has its own nervous system called the enteric nervous system, which is sometimes referred to as the second brain. And the brain communicates with the enteric nervous system through the vagus nerve. So the vagus nerve carries information to and from the brain. And lack of vagus nerve stimulation can lead to poor digestion and constipation. Datis Karizian, if any of you have read his book, Why Isn't My Brain Working? Really useful book, talks about how to stimulate the vagus nerve and so improve gut and brain function, so by gargling, singing and activating the gag reflex. And he suggests that chronic gut problems are sometimes the first sign of brain degeneration. And then finally, we've got immunological, so cytokines, which form a route of communication between the intestines and the central nervous system. And they can enter through leaky regions of the blood-brain barrier. So poor brain function can compromise gut health, and vice versa. Poor gut function can compromise brain health. And the classic example of a condition where there's dysfunction of the brain-gut axis is IBS. So factors affecting the gut-brain axis, including the intestinal microbiome, leaky gut, and hypersensitivity to food allergens. And gluten is relevant to all of these. So firstly, the intestinal microbiome can be altered by gluten, and this is important in relation to brain health because the gut flora play a key role in the central nervous system. So for example, the gut flora affects the production of brain peptides like BDNF, brain-derived neurotrophic factor. That's sometimes called miracle growth of the brain. It's involved in brain development and plasticity. Plasticity refers to the ability of the brain to strengthen and form new connections, and contrary to what we used to think, this can occur throughout life. So the brain remains plastic, and BDNF is really important for that. Also, neurotransmitters are secreted by the gut flora, so serotonin, GABA, catecholamines and histamine. And gut microbes can also affect the production of inflammatory and anti-inflammatory cytokines. So leaky gut, gluten can obviously, as we've already heard, cause leaky gut. As we heard from Eve, an enzyme in the gut called zonulin has an important role in regulating gut permeability, and increased expression of zonulin results in increased permeability. So gluten triggers an increase in zonulin, which can cause the destruction of the tight junctions, allowing antigens, for example LPS from bacteria, so lipopolysaccharide from bacteria, into the body, and this can trigger systemic inflammation, which can ultimately change brain chemistry. And also, zonulin has been found in the brain, which I'll mention on a further slide. And Alessio Fasano was the researcher who discovered zonulin, and he explains how this leakiness occurs in pretty much everyone when they eat gluten. So regardless of whether you've got gluten sensitivity, when you eat gluten, it will trigger increased expression of zonulin, but some people will be able to restore the integrity of the gut barrier quickly after eating gluten and deal with the inflammation, and other people won't. They'll have ongoing leaky gut, and this is when problems occur, when increased antigens continually entering the circulatory system and activating an immune response. And then finally, we've got hypersensitivity to food antigens, including gluten. And of course, these three factors can affect each other. So for example, the gut microbiota obviously can also lead to leaky gut. This paper reviews research showing that gluten sensitivity can harm the brain and nervous system, leading to a number of different neurological problems. And he basically says that gluten sensitivity destroys the brain and nervous tissue more than any other tissue in the body. In fact, even more than that of the gastrointestinal tract. So, in other words, that the most common area of non-intestinal manifestation of gluten sensitivity is in the brain and nervous system. And there are various estimates of how many people may suffer gluten sensitivity. Immunologist Dr. Bajani has suggested that it may be as high as 30% in the general population, and probably the people that we see in practice is going to be a lot higher than this, because obviously we're seeing people with health issues. The blood-brain barrier is similar in structure to the intestinal barrier. It consists of endothelial cells with tight junctions which restrict diffusion of water-soluble substances from the blood to the brain. So, for example, a healthy blood-brain barrier prevents pathogens and other undesirables from getting into the brain. And there appear to be a number of mechanisms involved in gluten's assault on the brain and the nervous system. So, firstly, zonulin, which we've already mentioned. zonulin receptors have also been found in the brain, and overexpression of zonulin could be involved in blood-brain barrier disruption in a similar way to how it increases the intestinal permeability. So Alessio Fasano's research reveals that the same mechanism by which gluten increases gut permeability also leads to a breakdown of the blood-brain barrier. Then we've got the anti-gliadin antibodies, which can react with brain proteins, causing a breakdown of the blood-brain barrier. They, in any case, result in increased production of inflammatory cytokines like TNF-alpha and activation of the COX-2 enzyme, which are key players in neurodegenerative diseases. And of course, people can have a double whammy, so they might have SNPs that predispose to overexpression of TNF-alpha or IL-6. And I think the myDNAhealth comprehensive test does include the TNF-alpha reports on that as well. Anti-gliadin antibodies also appear to cross-react with certain brain proteins that look like gliadin. So, for example, they bind to a protein called synapsin-1, which is involved in synapse regeneration. And the authors of the study that found this said that this may explain why gliadin contributes to neurologic complications such as neuropathy, ataxia, and others. In the brain, gluten can trigger immune reactivity to transglutaminase 6. We heard earlier about TG2. Well, TG6 is the version of this that is found throughout the nervous system, and this can lead to autoimmune destruction of brain and nervous tissue. It can alter the gut microbiota, resulting in dysbiosis, as I mentioned before, and this can disrupt the gut-brain axis, firstly by increasing LPS and systemic inflammation, and also, of course, alterations in the gut microbiota can lead to alterations in the production of short-chain fatty acids, which play a critical role in maintaining the intestinal lining, but also butyric acid can cross the blood-brain barrier and has a role in gene expression. And evidence from germ-free mice suggests that the microbiota can affect the blood-brain barrier, and that this might be due to, or the mechanism might be due to short-chain fatty acids produced. A very obvious mechanism by which gluten can affect the brain, of course, is that malabsorption of nutrients may occur if there's problems with the gut, and this can obviously have impacts on the brain and nervous system. And finally, the opioid effect of gluten is relevant for some people. So incomplete digestion of gluten results in gluteomorphins, which are peptides that can cross the blood-brain barrier and have an opioid effect on the brain. And this is one of the reasons that some people, I don't know if you've had this in practice when you've asked people to give up gluten and they've had unpleasant withdrawal symptoms like low mood and apathy when they stop eating foods containing gluten. And gluteomorphins also play a role in behavioural problems and mental health problems. And the production of these is thought to be related to low levels of a protease enzyme called DPP. And these enzymes actually break down gluteomorphins in the gut, but if there's a lack of them, then it can lead to this problem in the brain. So I've already mentioned that gluten can disturb the gut flora. And as you know, there are lots of other factors as well. I'm not going to go through these, you're all very familiar with them. But gluten-containing diets can basically lead to a reduced diversity in gut flora compared to gluten-free diets. Not quite sure why this is. Maybe it's just that some bacteria don't like it, or perhaps it's because it triggers leaky gut, which then impacts on the gut flora. And this paper, entitled Non-celiac gluten sensitivity triggers gut dysbiosis, neuroinflammation, gut-brain axis dysfunction, and vulnerability for dementia. So this links the change in gut flora to upregulated inflammation, which I've been talking about due to LPS and synthesis of pro-inflammatory cytokines. And this increased inflammation becomes more of a problem with age, because as we age, a chronic low-grade inflammation occurs anyway. So as we age, we will tend towards increased levels of TNF-alpha, IL-6, and CRP. and it's likely that a change in the composition of the gut microbiota during aging plays a role in the chronic activation of the immune system and consequently chronic inflammation. And another study found that frail older people have a different microbiota compared to healthy older people. This paper suggests that Alzheimer's may actually begin in the gut and be related to an imbalance of gut microbiota. And Alzheimer's, of course, is a particular concern because dementia, as of November last year, dementia became the leading cause of death in the UK, and numbers of people with dementia are predicted to increase fivefold by 2050. And this one links leaky blood-brain barrier to Alzheimer's, and we've discussed the ways in which gluten can affect the blood-brain barrier. And then other neurological conditions, a few examples here. Patients with neurological disease of unknown cause were found to have a much higher prevalence of circulating anti-gliadin antibodies, so 57%, in their blood than either healthy control subjects, which had 12%, or those with neurological disorders of known etiology or cause. And the first author on this paper, Haji Batiliu, is a UK neurologist, and he's been, I'm sure you've heard of him, at the forefront of research linking gluten to neurological disorders, in particular ataxia. Also, in relation to migraine or headache, so high prevalence of headache and migraine has been found in celiac patients compared to a control group. And Dr. Haji Batiliu, again, he's demonstrated that a gluten-free diet can result in complete resolution of headaches in people with gluten sensitivity. Other neurological conditions, anxiety, depression, bipolar, schizophrenia, epilepsy, some forms of ataxia, autism, ADHD, MS, Parkinson's and others, as well as, of course, symptoms such as brain fog, poor concentration. So, in summary, gluten can affect the brain through multiple mechanisms, contributing to multiple neurological conditions. There are effects on increasing systemic inflammation, degrading the blood-brain barrier, altering neurochemicals in the gut, and triggering autoimmune reactions. And some authors suggest the effects of gluten on the brain may be more significant than on any other area of the body. Thank you. That's it. I'll just switch off so Tony can come back in.
Thank you so much, Claire. That was really interesting. Just a couple of questions. First one, what nutritional protocols do you recommend to treat gut inflammation?
Okay, so can you hear me again? Yeah, so obviously first we all know about things like the 4R program, which would be appropriate for this. And I'm sure everyone's very familiar with the elements of that, but just to recap, well, sometimes now it's called 5R, of course, but it refers to remove, replace, re-inoculate, repair, and the 5th R is rebalance. So remove refers to identifying any triggers to gut inflammation, which would of course include gluten, but other foods as well, and also removing any dysbiosis of bacteria, so antimicrobials might be relevant, like oregano, garlic, folic acid. Replace refers to adding in digestive enzymes and hydrochloric acid if appropriate. The 3rd R, re-inoculate, refers to including probiotics, which can help with gut repair by stimulating the production of short-chain fatty acids. And also they stimulate the gut's immune system and compete with undesirable organisms, so can help rebalance the gut flora. And then nutrients to repair the gut. So there's quite a few of these that are important. Vitamins A, D, zinc, the B vitamins for methylation. So I would suggest definitely an all-round multivitamin and mineral, and perhaps for some people extra vitamins A and D. Did I say zinc? Zinc is important, of course, for protein synthesis, so for cell repair and also for immune system. Curcumin is often used as an anti-inflammatory as part of gut repair programs, and also L-glutamine, which is used as a fuel by the gut cells. although there is some concern about using this with candida overgrowth, so there's just a caution there. And of course, essential fatty acids are really important. There are other supplements that can be helpful as well, but obviously the precise recommendations would need to be individualised to the client. And the other thing to mention, of course, is bone broth, which is great for gut healing, but needs to be made with organic bones.
Thank you, Claire. That was a really detailed, comprehensive answer. Second question. One of the practitioners has a patient who's been tested with myDNAhealth, and the patient has the APOE4 risk gene, which is associated with Alzheimer's. Should they remove gluten from their patient's diet?
Okay, I'm assuming from that they haven't tested high risk for celiac, as well. But obviously ApoE4, so that increases the risk of Alzheimer's, and it codes for apolipoprotein, which is involved in fat transport. But Professor Bredston and his team published a paper last year explaining that ApoE4 also acts as a transcription factor. And this is for about 1,700 genes, of which about 76 have a role in Alzheimer's. So these would include genes that increase inflammation, lead to insulin resistance, etc. So given that people with ApoE4 have an increased tendency towards inflammation, and that gluten increases inflammation, that would certainly be a reason to be proactive about removing gluten from the diet. Certainly if the person is in poor health. I guess if somebody has taken the test just out of interest that they're in good health, then they may at least want to reduce their intake of gluten. Because we are actually eating quite a lot of it. In Europe, the average consumption of gluten is 10 grams to 20 grams a day, and some people eat as much as 50. So all individuals, even those at low risk, could have some form of gluten reaction at some point in their lives. So should perhaps look to reduce it. Obviously, it's very easy to choose alternatives at breakfast and supper. Lunch is a bit more challenging, but it would be advisable, I'd say, for anyone with ApoE4, even if they're in good health, to avoid it at breakfast and supper, and eat it as little as often as lunch. But obviously, for those who have a reaction, then 100% avoidance is necessary.
Thank you, Claire. With time now, I've got a final comment or question I'm going to throw out to both presenters, Eve and Claire, and it's I think it's following on from what you were just talking about there, Claire. So it goes back to, or if you can make a comment about this, does the quality of the food containing gluten matter? So in other words, should we remove gluten from our diet, or should we actually look at the quality of the food that actually containing gluten and base some of that decision on that in addition to the genetics? If you can maybe, sorry, Claire, if you want to try and answer that first, and then hopefully you can add something to it. Thank you.
Yeah, I think if somebody has already developed a sensitivity to gluten, then it doesn't matter what the food is. They need to have 100% elimination, and that includes looking at all areas of contamination, things like soy sauce and other things that can have very small levels of gluten in. If somebody doesn't have a sensitivity to gluten, then certainly eating bread, if from day one when we were born we all ate a 4-day fermented rye bread with lower levels of gluten that had been properly fermented, and we didn't eat too much of it, probably most people wouldn't develop a problem. So certainly for those who haven't already developed a problem with it, that would be the advice, to eat something that has had a longer ferment, certainly than the 10 minutes, which I think most bread has, and just not eating too much of it, and eating things like rye bread or spelt with lower levels of gluten.
Yeah, absolutely, Claire. Brilliant answer. I agree wholeheartedly. But with regards to the DNA testing, so at DNAhealth, there's no there's no evidence for the quality of food with relation to genetics. I mean, it's an interesting question, but it goes back to what I said previously about the non-celiac gluten sensitivity. There's very little known about it, but I think we know much more within nutritional therapy rather than genetics. But this is an area that we'll just keep looking at and reviewing to see if there's some clues that can help us on the DNA side.
Thank you. Thank you to both Claire and to Eve. I think it was a really informative webinar this evening. We will be sending out the recording, so apologize for some of you experienced poor connectivity and sounding issues. The recording should be fine, and we'll also send out a handout, and we'll send out a copy of all the questions and the answers that have been posed as well. So thank you again for everyone attending. A huge thank you to our two great presenters this evening as well. Thank you and have a great evening.